bilgilendirme olursa inşallah fırsatım olursa gelmek isterim ama
Tabii
online
ki
oldu.
hocam.
Evet. Oldu hocam. Görüşmek üzere.
Sağ olun. Arzu ederseniz devam edebilirsiniz hocam. Şu anda da son oturumumuza geçiyoruz.
Ha, olabilir hocam. Kalabilirim. Kendimi sessize alayım.
Şimdi
Görüşmek üzere
sıkışık
hocam.
olduğu için burada hiç ara vermeden başlıyoruz. Ee, ama Parwiz, Niazi, Abdul, Bari, Hejran. I think they are not here. The second one, Neziri Göç, is there anyone from Bayakı, Özgentürk? Salisu is here. Emine, Erdağ, Akca is here. I am here. Okay. Yes, welcome everybody. This is the final session of our Congress, session 30. I am Mehmet Çevik from İzmir Katip Çelebi University, and I'm the chair of this session also. In this session, we have normally 7 presentations. Maybe the number may change. So we can start with the first one. I think the first 2 presenters are not here. We may start with Salisu. Salisu.
Okay, sir.
Yes, please.
Okay, I hear you, sir. Good afternoon. Hello, sir.
Yes, we can see your presentation now.
Okay, sir. Good afternoon. How are you? I'm Salisu. I'm here to present my research, which titled Optimized Basic Assisted Extraction and GCMS Analysis of Bioactive Compounds in Mango and Neem leaves, prospect for pharmaceutical and biofuel application. I am from the Department of Bio and Industrial Chemistry, Umar Musa Adwa University, Nigeria. Introduction. Optimized basic assisted extraction. Hello, sir.
Can you make it full screen, the presentation?
Okay, full screen.
F5 or...
It's okay like that. Hello, sir.
Top menu, slideshow menu. Top slideshow menu. You may select full screen, or this is okay.
This is okay for me.
From beginning, the 4 left one.
This is okay like that.
Yes, that's very good.
Okay, sir. Introduction. Optimized basic extraction is a chemical process that uses a basic alkaline substance like sodium hydroxide to improve the efficiency of the extraction valuable compound from mango and neem leaves. Then GCMS analysis, which is called gas chromatographic mass spectroscopy, is a technique used to identify and analyze specific bioactive compound present in the extracted plants. Then bioactive compound are naturally occurring chemicals in leaves that have medicinal and industrial application, such as antioxidants, anti-inflammatories, or antimicrobial activities. When we talk about prospect for pharmaceutical and biofuel application, this study explores how the identified compound can be used in pharmaceuticals. Example for medicinal plants are vital to healthcare, especially in developing countries, where up to 80% of people depend on them. When we talk about medicinal plant, like the ancient time, we use plant to treat the diseases. Then we talk about the mango. We know that mango leaves contain mangiferin and phenolic compound, which contain antioxidant, antibiotic, and their wound healing properties. Then neem leaves, which is called Azadirachta indica, are rich in terpenoids, flavonoids, and fatty acids. They are useful in treating infections and the infectious diseases, such as chronic diseases like cancer, AIDS, etc. Then this study focused on optimizing alkali assisted extraction and analyzing the bioactive compound using GCMS, aiming to support pharmaceutical and the biofuel application from these plant resources. Then this is my methodology. This is sample collection and preparation. I collect the fresh and mango leaves are collected from Mashi within Nigeria. The leaves were washed with distilled water to remove dirty and shed dried for 3 days to maintain their phytochemical integrity. Once dried, the leaves were ground into fine powders using mortar and pestle and stored in a tight containers at room temperature to prevent contamination. Then the procedure, 20 grams of powdered leaf sample of both the neem leaves and mango leaves was dissolved in 30 ml of distilled water with the ratio of 0.5 grams of sodium hydroxide, 1.0 grams of sodium hydroxide, and 1.5 grams of sodium hydroxide. Here is the ratio different. We check the difference of the ratio of the catalyst, which is sodium hydroxide. Then the solution was treated at 100 degrees Celsius for about 30 minutes using a hot plate magnetic stirrer. Then the extracts were filtered using Wattman filter paper and stored in a level sample bottle. Then we observe the sample that contains 1.5 grams of sodium hydroxide has more yield of extraction. Then we take it to subject the gas chromatographic analysis. This is the schematic diagram for the extraction process for mango, which is the fresh mango leaves. This is the dry mango, then powdered mango leaves, extraction process, filtration process, and then the filtered mango leaves, which is ready to do an analysis. This is a schematic diagram designed for extraction process of neem, which is Azadirachta indica leaves, fresh neem leaves, dried neem leaves, the powdered neem leaves after grinding, then the extraction process, filtration process, and the filtered neem leaves. This is the results that found on GCM analysis, identified compound GCM analysis of mango leaves. We found that in neem's leaves, we have 27 compound present in it. This is a compound name, molecular formula, it is molecular weight, height, percentage, and biological activities. Then we know these are the 27 compounds that are found in mango leaves. Then the compound identified from GCM analysis of neem, Azadirachta indica leaves, were found to be 20. Then the extract of mango leaves identified 27 bioactive compounds with varying molecular weight. But the major constraint detected in mango leaves include fatty acids, esters, and sterols, which contribute to the antioxidants, anti-inflammatory, antimicrobial, and in cosmetic application. Then for the extract of neem leaves contain 20 bioactive compounds that exhibit antimicrobial, antifungal, and antioxidant property. Then the most abundant compound in neem leaves include terpenoids, flavonoids, and fatty acids, which contribute to neem wide-ranging medicinal, and industrial application. Then these are the structure of the major chemical compound extract from both plants, neem and the mango leaf. We have 1,3-cyclobutadiene 2,2,4,4-tetramethyl, 9,12,15 octadecatrinoic acid methyl ester, hexadecanoic acid methyl ester, 2-nonoic acid methyl ester, pentadecanoic acid methyl ester, 912 octadecaniol, niol chloride, ZZ 912 octadecanoic acid. Then this study highlights the significant potential of neem and mango leaves as suitable sources of bioactive with diverse application in both pharmaceutical and industrial application. Then the use of sodium hydroxide as a catalyst enhances the extraction process to enable the recovery of high-value compound. Then this research contributes to the growing body of knowledge supporting the stable utilization of plant-based resources. This is my reference. Thank you for listening.
Thank you for the presentation, Salisu.
Okay, sir. Thank you, sir, for listening.
Are there any questions about the presentation about the neem and mango leaves?
Neem and mango leaves.
Yes.
Can I
Yes,
I-
please.
can I ask one question if possible?
Sure.
Okay.
In your presentation, you mentioned about the effect of sodium hydroxide as the extraction aid. So in your next studies, I mean, why did you use sodium hydroxide? And would you advise any other chemicals or biogreen methods for the extraction of bioactives from these mango and neem leaves? I just wondered, why did you use sodium hydroxide?
Because we use it to it gives us the more yields of the extraction from the plants compared to the other solvents.
So you used other solvents and you compared the efficiency. Okay. Okay. Thank you.
Okay.
Any other questions? Salisu, what is the origin of this plant? Neem.
Origin. Neem.
Yes.
Origin.
Origin African or
It's from
English?
Nigeria. From Nigeria.
Nigeria. Yes. Have you been using Salisu leaves for
Salisu
centuries?
Neem. What do you say?
Have you been using neem leaves for centuries, for many years?
Yes.
Yes. Okay. Thank you.
Okay.
Any other questions? Then let's continue with the next presentation. I think Emine Erdağ Akca is here.
İkinci sırada ben varım da beni geçtiniz az önce. Sunuma da başladığınız için bir şey demek istemedim.
Tamam. Başta bir anons yaptım ama isim herhalde şey oldu. Özür dilerim. Sizinle devam edelim. MB 2503 evet. Bilge İrem Göç.
Evet.
You may start your presentation. Can you also turn on your camera? Bilge? Okay, thank you.
Hello everyone, my name is Bilge and I will be present our research titled Multi-Omic Integrated Analysis of Endometriosis and Autoimmune Diseases, Identification of Novel Biomarkers. Endometriosis is a chronic estrogen-dependent inflammatory disease that affects around 10% of reproductive age women worldwide. It is characterized by the presence of
why do you use sodium hydroxide?
The presence of endometrial-like tissue outside of the uterus, often in the ovaries, fallopian tubes, and peritoneal cavity. Despite being traditionally viewed as a gynecological disease, recent studies have highlighted that the role of immune dysregulation in its pathology. Yet the pathology of endometriosis is not fully known. Autoimmune disorders are marked by dysregulated immune response against the self tissues. Interestingly, the endometriosis shares several clinical and molecular features with autoimmune disease, like chronic inflammation, immune cell infiltration, and aberrant gene expression. But a comparison between these diseases is still lacking in the literature. In this study, we aim to perform a multi-omics data integration between these disorders, including transcriptomic and proteomic data. The aim is in order to identify dysregulation genes of both disease, common genes, explore common biological pathways, and reveal the key regulatory genes on potential biomarkers of between these two diseases. In the methods we used, we first collected the data from publicly available data sets. We use Geo and Array Express to retrieve data for transcriptomics, and additional proteomics insights were drawn from the literature-based research. For transcriptomic analysis, we conducted differential gene expression analysis using standard pipelines in our studio, like Lima and DESEC, depending on the platform type of the dataset. After that, we focused on the finding commonly up and down-regulated genes between endometriosis and autoimmune diseases, such as systemic lupus, rheumatoid arthritis, multiple sclerosis, etc. Then we performed gene ontology, KEGG, and reactome pathway analysis, enrichment using R packages for that bioconductor. After that, protein-protein interaction networks constructed via string in cytoscape to find hub genes. And last, XL infiltration were made by using R Studio again to estimate immune cell infiltration levels across the conditions, controls and case dependent. After that, in the result, we identified 1629 commonly upregulated genes and 1162 downregulated genes shared between these both diseases. And we used the cytoscape to most frequent genes to show the interaction between them. These are the most upregulated genes, and these are the most downregulated genes. For example, some of the up and down-regulated genes like VCAM1, LI96, ASR1, and ERBB3 are involved in the process like immune activation, signaling and hormone response. Also, these genes are the potential key regulators at the intersection of immune and hormone signaling of this protein-protein interaction results. In enrichment analysis, upregulated genes were significantly enriched in inflammatory response, chemokine, and toll-like receptor signaling. And NFKB and interferon signaling. Downregulated genes were involved in estrogen response, epithelial development, and cell adhesions and junction organization. These results support that a dual dysregulation both of immune and hormonal pathways. After that, we use the X cell infiltration to detect how these two conditions are. We say to immune cells to define the levels of immune cells according to conditions. We found the increased infiltration of CD8 positive T cells, Th1 and Th17 cells, and dendritic cells. Also, these are suggest the a shared pro-inflammatory immune environment. Interestingly, some of the B cells were downregulated in the autoimmune diseases. When we came to the conclusion, our findings demonstrate the endometriosis and autoimmune disease shares significant molecular overlap, particularly in immune-related and hormone-regulated pathways. Upregulated genes were mostly associated with immune activation. Downregulated genes indicated impaired epithelial and hormone pathways. According to these results, the idea of endometriosis may exhibit autoimmune-like behaviors that shared hub genes may serve as a potential biomarker and therapeutic agents. After that, moving forward, our future directions include experimental validation of the identified genes and pathways using techniques identified genes such as qPCR, Western blot, etc. Or we can move the clinical evaluation in patients' cohorts to assess their diagnostics or prognostics values, or integration of additional omic layers such as DNA methylation, single-cell transcriptomics, and histone marks. That's all. If you have any questions, feel free to ask.
Thank you for the presentation. Are there any questions to Bilge? Bilge, are you a master student? Bilge, can you hear me?
Yes.
But I cannot hear you. Ses gelmiyor. Bilge çok derine gitti. Şimdi hiç gelmiyor. Mikrofon uzak galiba. Kapandı, açıldı. Tamam. Teşekkür ederiz. Soru var mı başka? Soru yoksa Emine, Erdağ, Akca ile devam edebiliriz. Burada kendisi karotenoidlerin püskürterek kurutulmasına kullanılan duvar materyalleri. Buyurun. Sizin de ses gelmiyor. Bende mi sorun acaba? Oradan ses geliyor mu? Benim sesimi duyabiliyor musunuz? Bilge hocam veya Bilge, Emine, Erdağ Hanım'ın sesini duyabiliyor musunuz? Mikrofonu kapattınız şimdi.
Sizi duyabiliyorum ama duyamıyorum.
Emine hocam konuşur musunuz? Ses geliyor mu? Bilge duyabiliyor musun?
Hocam ben duyamıyorum. Ben duyamıyorum.
Biz de duyamıyoruz.
Emine. Nerede? Tekrar. Sizi tekrar çıkarıp geri alsak olur mu Emine
Evet.
Hanım'ı?
İsterseniz bir ayrılın, tekrar bağlanın. Hala gelmiyor ses. Ee, Emine hocam bir ayrılın, tekrar bağlanın. Bu arada benim de bir sunumum var. Ben onu yapayım. Siz tekrar bağlanınca yeniden başlatırız. Yes, dear participants, I also have a presentation today related with artificial neural networks applied in mathematics, physics-informed neural networks, abbreviated as PINs, for solving differential equations. The outline of my presentation is including an introduction. I will overview the PINs, solve a problem of one-dimensional heat equation as a example test problem. I will mention the advantages and challenges and then conclude. First of all, let's mention about simply differential equations. They are the foundation for modeling physical systems in disciplines ranging from fluid dynamics and quantum mechanics to structural analysis and biomedical engineering. In fact, in all areas, engineering areas, we use models and mathematically they are represented with differential equations. So, accurately solving these equations is essential for understanding and predicting real-world phenomena. Ordinary and partial differential equations can be solved using various approaches. Therefore, the differential equations are mainly classified as ordinary or partial according to the number of unknowns or and various approaches. We use various approaches depending on their type and complexity. What are these main approaches? For many centuries, even centuries, 200 or 300 years, mathematicians developed methods. The main methods are analytical methods such as separation of variables, integrating factor, characteristic equation, etc. There are numerical methods like Euler's method and Runge-Kutta or finite element or finite difference or spectral and collocation methods. With the development of computers, these numerical methods became more popular and used in the industry also. There are graphical methods like Lyapunov's method or phase plane analysis. These are not very common. And there are some semi-analytical methods like homotopy analysis, homotopy perturbation, adomine decomposition, and perturbation methods. These are the main approaches. However, with the invention of artificial intelligence, people started to implement artificial intelligence to solving differential equations, because the classical methods have been a standard tool, but they have also some challenges, and we try to apply artificial intelligence to everything. So we also try to use it in mathematics for the solution of differential equations. So recent advances in artificial intelligence, especially for the last 4 or 5 years, even 3 years, have led to the development of alternative data-driven approaches. These AI-based, artificial intelligence-based methods aim to offer greater adaptability, improved scalability, and in some cases enhanced accuracy compared to conventional techniques. They have advantages and disadvantages. The most notable AI approaches are physics-informed neural networks that I will mention today. Other approaches are deep Galerkin, neural ordinary differential equations, deep Ritz approach, deep backward stochastic differential equation, reinforcement learning-based techniques, generative adversarial networks, hybrid AI numerical methods, and some others also. This method pins were first introduced by a paper published in 2019. Even the name was first used in that publication by Raisi, Perdikaris, and Karniadakis. Unlike the traditional neural networks, in fact, neural networks were used previously as a method and approach of artificial intelligence. The difference is this approach is physics informed. I will mention about it. Pins are designed to respect the underlying physics of the problem by embedding the government equations directly into the training process. This innovative approach enables the network to learn solutions that satisfy both data, if available. If you have, for example, experimental data, you may also embed the data and the underlying physical laws simultaneously, which are the differential equations themselves. The general architecture of a pin is something like this. Classically, in neural networks, we had input, a network, and an output. But in a pin, we also include some other data, which is related with the differential equation itself. And we calculate a kind of error. We call it loss, and we try to decrease or minimize that loss. So a typical pin is a deep neural network that takes coordinates as inputs, for example, X, Y, Z, T, like that, and outputs the predicted values of the solution, for example, temperature, velocity, displacement, or pressure. The network is trained to minimize a loss function. This is the typical term used in pins, loss function, that includes not only discrepancies from data, but also penalties for violating the governing partial differential equations and boundary and initial conditions. What is a loss function, by the way? A total loss function is a loss function that combines multiple terms to balance different objectives during training. These multiple terms are, the first one is related to partial differential equation or the differential equation itself. So the solution has to satisfy the equation. The solution has to satisfy boundary and initial conditions. So we have a loss term related with boundary and initial condition. And finally, there might be a loss related with available observational or experimental data. So each of them has some contribution denoted by lambda here. Three components of the loss. So the main idea is that the network is trained to minimize the total loss. I continue with an example, simple example problem. This is a simple differential equation of one-dimensional heat equation. We don't use any data. We only use the differential equation. As mentioned here, del U over del T is equal to alpha times del squared U divided by del X squared. Here U is the temperature at position X and T. Alpha is the thermal diffusivity. We have boundary and initial conditions as given here. U of 0 and T and U of 1 and T are equal to 0. This means that X0 and X1, since the length is 1, are the ends of the rod. At the ends, the temperature is 0. We consider a time interval between 0 and 5. Initial condition is given also here as sine pi x. The steps are for solving the problem or finding a solution. We define a neural network solution, which is denoted as U of theta with inputs X and T, and the output is U, the temperature. And the algorithm calculates automatically the partial derivatives of the differential equation, first derivative and second derivative, using its embedded libraries, then construct a residual function, which is given here. The equation was del U over del T is equal to this term. But we define the residual as the difference of these terms. And if we find a zero residual, then it means the equation is satisfied. The residual function represents the error in satisfying the differential equation. And in the fourth step, minimize the mean squared error of the residual at collocation points. We select some collocation points, ensuring that the neural network output satisfies the equation. Then we enforce the initial and boundary conditions. Also, sorry, some people are coming in. The function, the solution function must satisfy the heat equation, and the pin learns a function that approximates the analytic solution. This is the known analytical solution. So the best solution will be equal to this one. So we made these steps just by using ChatGPT. ChatGPT prepared the Python code for us, and we copied the Python code and run the code in Colab, also Google's Python compiler. And we obtained this result. This is a solution. In X, as you can see, the length is from 0 to 1, and the time was from 0 to 5. And this is the solution, pin solution for the one-dimensional heat equation. So as you can see it, or we can understand, but you can also understand this is a logical or expected solution. So in this solution, we used no experimental data. We just used the differential equation and the boundary conditions. This highlights the capability of PIN's method to solve complex physical problems. This is a simple problem, but we start with these simple problems. Probably in a couple of years, we will continue with more complex problems. What are the advantages of this method? It's mesh-free. So this is an advantage. Unlike conventional numerical solvers such as finite element or finite difference method, they are mesh dependent. In pins, we do not require discretization. Integration of prior physical knowledge is another advantage. Pins leverage partial differential equations and governing physical laws as constraints within their loss function. Unified representation of forward and inverse problems. It's a key advantage of PIN, is that its ability to solve both forward and inverse problems within a single neural network. Improved generalization and extrapolation capabilities, unlike again traditional machine learning models, which predominantly rely on empirical data. PIN incorporate physics-based constraints. In fact, I want to mention that classically we used, by classically I mean, for example, for 30 or 40 years, we are using neural networks. For example, when I was studying preparing my PhD thesis before the year 2000, I used neural networks as part of my thesis. That means about 30 years ago it was used. But in those times, we had numerical input data and just output. But today the difference is we also implement the differential equation in the solution neural network. This is the main difference with the classical or traditional machine learning models. Multi-physics and multi-scales integration. In fact, this is also valid. This advantage is valid for many other numerical methods like finite element. But there are also challenges. No method is perfect. So every method has advantages, but they also have challenges. The computational complexity and training cost is one of the challenges. Minimizing a total loss function is computationally demanding, especially in high-dimensional problems. Hyperparameter sensitivity and optimization challenges. Improper tuning of hyperparameters, which are network architecture, activation functions, and weighting factors, can lead to poor convergence. In fact, this is a challenge of neural networks. If you start with an improper activation function, you may not reach a solution. So you have to start somewhere which is close, maybe, or which will bring you to the solution. Otherwise, you will face numerical instabilities. It's also difficult in handling stiff equations. For example, smooth functions and continuous functions, PIN method is really advantageous. But if there are discontinuities, you may face problems. And also, we have challenges in scaling to large scale and high dimensional systems, and interpretability and robustness in real world applications is also a challenge. When you try to apply any method to real world problems, you will face some problems. And as a conclusion, we may say that PIN represents a significant advancement in the intersection of scientific computing and machine learning. Physical laws are embedded, and this is a main advantage. We demonstrate the effectiveness with a test problem. And despite their promising capabilities, PINs also face challenges related to computational cost, optimization, complexity, and scalability, particularly in high-dimensional or stiff systems. As the field matures, PINs are poised to become a core tool for tackling complex real problems across physics, engineering, and applied sciences. Thank you for your patience. I may take questions if there are any questions. Bilgi hocam, sizin alan makine mühendisliği miydi?
Hocam benim alan gıda mühendisliği. Gıda
Nasıl?
mühendisiyim. Gıda mühendisiyim hocam.
Gıda.
Hı hı.
Sizde de diferansiyel denklemler var tabii.
Var hocam, var. Evet.
Ama tabii herkes matematik kısmıyla ilgilenmiyor.
Evet.
E,
Yani
Burada da işte gördüğünüz gibi yapay zeka her yere uygulanıyor. Dün kongremizin açılış programında davetli konuşmacımız vardı, Yusuf Sinan hocamız.
hı hı.
Yapay zekanın eğitim alanına uygulanmasından bahsetti. Çok ilginç, orijinal şeyler söyledi. Biz de istifade ettik. İşte matematikte diferansiyel denklem çözümüne de uygulanıyor. Muhakkak sizin alanlarınızda da bir yerlere uygulanıyor. Hatta hocamız şöyle bir şey söyledi dün. Yapay zeka herkesin kullanması lazım. Kullanmayan bir süre sonra geride kalacak.
Evet,
Bu
evet
kadar
hocam.
net.
Çok net.
Başka soru var mıdır acaba? Evet, ben o zaman tamamlamış oldum.
Teşekkür ederiz hocam.
Emine hocamı tekrar alıyoruz.
Hocam sesimi bir tekrar deniyorum
Şimdi
ama
geliyor
geliyor mu?
ses. Tamam, şimdi çok güzel.
Sevindim. Bir de paylaşım yapayım hocam. Şimdi
Lütfen.
Hello, do you hear me? I suppose that the next
Hello,
presentation is
greeting.
mine.
Who is speaking,
Yes.
please?
From Çevik.
Yes,
Yeah, we are ready to present our presentation.
Abdul Bari, are you going to present?
I'm going to
Hello.
present.
Yeah. We are going to present.
Mhm.
Then we can continue with you because we waited for you at the beginning. And now she will, Emine, Professor Emine Erdağ will present. After her presentation, we will listen to you, okay?
Okay.
sanırım ekranımı görüyorsunuz hocam.
Evet,
So, yes,
tam ekranı çevirebilirseniz seviniriz.
Hı hı. Tamamdır. Herkese tekrardan merhabalar. Ben Emine Erdağ Akca, Manisa Celal Bayar Üniversitesi'nde doktora eğitimi görüyorum. Burada da sizinle bugün karotenoidlerin püskürterek kurutulmasında kullanılan duvar materyalleri ile ilgili bir çalışma paylaşmak istiyorum. Bu anlamda öncelikle püskürterek kurutma nedir? Buradan bir gireceğim. Ardından karotenoidler bize ne ifade ediyor? Bunların püskürterek kurutulmasına ya da herhangi bir şekilde kurutulmasına neden ihtiyaç duyuyoruz? Ve burada kullanılan duvar materyalleriyle ilgili birkaç literatürden çalışma verip bugünkü sunumu tamamlamayı planlıyorum. Kurutma, gıda, işte gıda takviyeleri, bioaktif bileşenlerin uzun süre saklanması için hem besin yoluyla hem de gıda içerisinde herhangi bir bileşenin alınması, ekstrakte edilmesi yoluyla uygulanan bir işlem aslında. Ancak burada kullanılacak olan materyal ve işlemin amacı da göz önüne alındığında başarılı bir sonuç almak için uygun bir kurutma tekniğini seçmek oldukça önemli. Günümüzde kurutma teknolojilerine baktığımız zaman özellikle besinsel bileşim, bioaktif bileşenleri kurutmada yaygın kullanılan işte spray dryer, püskürtmeli kurutucu, dondurarak kurutma dediğimiz freeze dryer prosesleri ve tabii ki gelişen teknolojiyle beraber bu her iki sistemin entegre edilmesiyle oluşturulan püskürterek ve dondurarak kurutma ikisinin de bir arada yer aldığı teknolojiler yaygın olarak kullanılmaktadır. Püskürterek kurutma dediğimiz şey aslında sıvı damlacıkların katı partikül formuna hızlıca dönüşümünü sağlayan tek aşamadan oluşan bir süreç. Burası tipik bir kurutma süresi ama burada kazanılan hızdan kazanılan girdi sayesinde bioaktif bileşenin daha iyi korunmasını sağlıyor. Sıvı çözelti sisteme beslendikten sonra sıcak havanın bulunduğu kurutma kabininde püskürtülüyor ve burada artan yüzey alanı sayesinde çok hızlı bir şekilde düşük nem içeriğine getiriliyor. Temelde aslında dört basamakta oluşuyor bu hızlıca gerçekleşen olay. İlk aşamada bir çözelti hazırlanıyor ve bu sisteme pompalanıyor. Sisteme pompalanan çözelti atomizasyon yani sprey şeklinde bir püskürtme ile sıcak havanın dolaştığı kurutma odasına geliyor. Bu noktadan sonra emisyon dehidre oluyor. Yani yüzey alanı artan damlacıklar, püskürtülmüş damlacıklar sıcak havayla katı parçacıklara dönüşüyor. Hocam benim sesim tekrar bana geliyor ama bende mi bir sıkıntı oldu? Şimdi daha iyi mi? Evet. Son aşamada ise düşük nem içeriğine gelen bu partiküller artık katı ürün olarak adlandırılıyor ve bir filtre ve toplama sistemi yardımıyla toplanıyor. Bitki hücreleri normal metabolik aktivitelerin bir parçası olarak bazı kimyasallar üretmekte. Bunlardan bazıları şeker ve yağlar gibi birincil metabolitler, bazıları da genellikle terapötik etkileri sahip bioaktif bileşenler olan ikincil metabolitler. Lipokramlar olarak da bildiğimiz karotenoidler bu ikincil metabolitler sınıfına dahil. Fotosentetik yolla bakteriler tarafından ya da fotosentetik olmayan yolla bazı mantarlar ve bitkiler, bakteriler tarafından üretimi, sentezlenmesi gerçekleştirilebiliyor. Bitkisel pigmentler olarak da biliniyor karotenoidler. Bitkisel pigmentler dediğimiz şey aslında az önce söylediğimiz bitkilerin ikincil metabolitleri. Aynı zamanda gıdalara bu renklerini kazandıran yapılar. Bunlar arasında karotenoidler yalnızca renk olarak değil, gıda maddesinin besinsel zenginleşmesine de katkıda bulunduğu için birçok araştırmaya sıklıkla konu olmuş durumda. Bununla birlikte çok çeşitli bir yayılım gösteriyorlar. Hem renk açısından farklı renk tonları, geniş renk yelpazesi, hem de doğada yaklaşık 850 farklı türü bulunuyor. Bunlardan yaklaşık %40'ını beslenme ile tükettiğimiz gıdalarda alabiliyoruz. Aslında çoğumuzun da belki duyduğu beta-karoten, likopen, lutein bizim bildiklerimizden başlıcaları. Karotenoidler antioksidan aktiviteleriyle bilinmektedirler. Ancak bununla birlikte birçok sistemik bozukluk, göz probleminde de yararlı etkileri söz konusudur. Yapılan çalışmalarda yüksek oranda karotenoidlere maruz kalan deneklerin en düşük konsantrasyonda tüketenlere kıyasla herhangi bir nedene bağlı ölüm oranında azalma gözlenmiştir. Benzer şekilde yine başka bir çalışmada yaşa bağlı hastalıkların da görülme sıklığını azalttığı görülmüştür. Bu tür etkileri aslında karotenoidlerin yüksek antioksidan aktivitesiyle ilişkilendirilmektedir. Ancak özellikle oksijene maruz kaldığında, ışığa ve ısıya maruz kaldığında, özellikle de bu gıda işleme proseslerinde bunların birçoğunu yaşaması veya depolama aşamasında bunları yaşaması sebebiyle yaklaşık %40'a kadar kayıplar yaşanabiliyor karotenoid içeriğinde. Bununla birlikte çevresel faktörlerin de dışında suda zayıf çözünürlükleri, düşük biyoyararlılığını da kullanımını, gıda endüstrisinde kullanımını oldukça kısıtlıyor. Karotenoidler bu söz konusu istenmeyen çevresel şartlara maruz bırakıldığında geçirdiği yapısal değişikliklerle beraber aslında biyolojik aktivitesi olmayan bileşenlere dönüşebiliyorlar. Karotenoidlerin püskürterek kurutulmasına geldiğimizde gelişen teknoloji ışığında yapılan araştırmalarla birlikte aslında püskürtmeli kurutucunun da kullanılan çok sayıda enkapsülasyon tekniğinden biri olduğunu görüyoruz. Püskürtmeli kurutucu aslında çok eskiye dayanan bir yöntem olmasına rağmen düşük maliyeti, sürekli çalışabilmesi, büyük ölçeklerde uygulanabilir olması, birçok duvar malzemesiyle uyumluluk göstermesi gibi nedenlerle hala en sık tercih edilen uygulamalardan birisi konumundadır. Ancak burada tabii ki çalışma koşullarının seçimiyle beraber duvar malzemesi de çok büyük öneme sahip. Çünkü bu kurutma esnasında sisteme verilen, yani bioaktif çözeltiyle beraber sisteme verilen kurutma malzemesi bioaktif bileşeni çevresel faktörlerden korumak için bir bariyer görevi görüyor. Bir matrix içerisinde bioaktif bileşeni hapsediyor ve çevresel etmenlere karşı bir koruyucu olarak yer alıyor. Bu yüzden kullanılan duvar materyalinin seçimi oldukça kritik. Püskürterek kurutma işlemleri karotenoidlerin stabilitesini arttırarak gıda endüstrisindeki kullanımını iyileştirebilir. Amaca ve sürece uygun duvar materyalinin seçimi bioaktif özelliğin sürdürülmesinin devamlılığı için kritik önem seviyesinde. Bu amaçla birçok çeşit birçok çeşit var kullanılabilecek. Karbonhidratlar, proteinler, lipidler veya hem bioaktif materyale hem de ortam şartlarına uygun bir şekilde el veriyorsa duvar malzemelerinin kombinasyonu da olabiliyor. Burada tabii ki çok çeşitli duvar materyalleri var ama ben literatürde özellikle çalışmalarda fazla kullanılanları, yaygın tercih edilenleri, pozitif ve negatif etkenleriyle almak istedim. Nişasta örnek verecek olursak polisakkaritler grubundan nişasta yüksek emulsifikasyon yeteneği olmadığı için düşük tutma kapasitesi yani bioaktif bileşenin düşük koruma kapasitesi gösterebiliyor. Maltodekstrin ve gamlar polisakkaritler sınıfından literatürde en sık kullanılan duvar materyallerinden. Özellikle maltodekstrinler tatsız ve çözelti halindeyken renksiz görünmesi sebebiyle özellikle gıda renklendirici karotenoidler gibi renk pigmentlerinin kapsüllenmesinde yaygın bir şekilde kullanılıyor. Bunun bir avantajı da yüksek giriş sıcaklıklarına bile dayanıyor olması. Bu da tabii ki elde edilen katı ürünün verimini iyileştiriyor. Kitozanlar da hem hidrofilik hem hidrofobik yani suda ve yağda çözünen bileşenlere uygulanabildiği için tercih ediliyor. Polisakkaritin diğer bir yaygın kullanan grubu gamlar olduğunu söylemiştik. Gamlar da kendi içerisinde sınıflandırılıyor. Ancak düşük su aktivitesinde bile uzun bir raf ömrü sağladığı için birçoğu yaygın olarak kullanılıyor. Duvar materyalleri de protein yapıda da olabiliyor. Protein yapılar içerisinde yine literatür çalışmalarında en fazla karşımıza çıkan peynir altı suyu proteinleri tek başına veya diğer duvar materyalleriyle kombine halde kullanılabilir. Bu peynir altı suyu proteininin yüksek bir film oluşturma kapasitesi var. Bu da kapsülleme sürecinde tabii ki besin maddelerinin etkin bir şekilde tutulmasını sağlıyor. Peynir altı suyu proteinleri dışında soya proteini de kullanılabilecek kaplama materyallerinden. Burada da biraz daha ayrıntılı çalışmalardan örnekler vermeye çalıştım. Karotenoid kaynağı olarak havuç işleme atıklarının kullanıldığı bir çalışmada peynir altı suyu proteinle inülin kombine olarak kullanılmış ve bunun düşük su aktivitesi, düşük nem içeriği ve daha küçük parçacık boyutu sağladığı yani daha iyi özellikler gösterdiği belirtilmiş. Bal kabağı çekirdekleri ve kabuğundan karotenoidler ekstrakte edilerek gam arabik duvar materyaliyle kaplanmış. Bunun da %79'luk bir tutma indeksiyle 90 günlük depolamada en düşük bozulmayı gösterdiği belirtiliyor çalışmada. Yine benzer şekilde karotenoidler guarana meyvesinden ekstrakte edildiğinde gam arabik duvar materyali olarak kullanılmış. Domates kabuklarından ekstraksiyonunu inceleyen bir çalışmada ise gam arabik ve maltodekstrin karışımı kullanılmış. Bu şekilde farklı sonuçlar gözlenmiştir. Bu sunu kapsamında şöyle sonuç olarak bir toparlayacak olursam renklendiricilerin ya da gıda renklerinin önemi tabii ki evrensel bazda doğal materyallere o artan ilgiyle beraber yapay renklendiricilerden doğallara bir alternatif arama arayışı yoğun bir odak noktası olmuştur. Bu anlamda özellikle karotenoidler yalnızca geniş yelpazedeki renk skalaları ile değil, potansiyel sağlık yararları ile de araştırmaların söz konusu odak noktası. Ancak yağda çözünmeleri, sulu ortama ilavelerini zorlaştırmaktadır. Düşük renk stabilitesi ve renk yoğunluğunun düşük olması, çevresel etmenlerde kararsız durumda olmaları gıda sistemlerine entegre edilmesini oldukça güçleştirmekte. Bu noktada en eski yöntemlerden de olsa basit olması, hızlı olması ve büyük ölçekli uygulanabilir olması nedeniyle püskürterek kurutma bir çözüm gibi görülebiliyor. Ancak işlem esnasında kullanılan duvar materyalinin seçimi oldukça kritik bu noktada. Hem biyoaktif bileşenleri hem çalışma koşullarınıza uygun olması lazım. Ki etkin bir bariyer görevi üstlenebilsin. Bu amaçla polisakkaritler, proteinler, lipidler veya bunların kombinasyonları kullanılabiliyor. Bunun dışında da geleneksel birçok duvar materyalinin dışında son zamanlarda yenilenebilir ve sürdürülebilir kaynaklardan elde edilen kapsülleme malzemeleri de araştırmalara söz konusu. Ek olarak da şunu söylemek istiyorum. Bu kapsül materyallerin evet yenilebilir, sürdürülebilir ya da işte dayanıklılık. Ancak şu anda birçok çalışmada da kontrollü ve hedefe yönelik salınımı ile ilgili. Yani özellikle mide koşullarının asiditesine dayanacak, bağırsaklarda salınımı gerçekleşecek vesaire. Yani bu hedef yer neresiyse orada biyoaktif bileşenin salınımına izin verecek duvar materyallerinin çalışılması ile ilgili de çok fazla ilgi söz konusu. Sunu esnasında kullandığım kaynaklar bu şekilde. Dinlediğiniz için tekrar teşekkür ediyorum. Soru varsa alabilirim.
Teşekkür ederiz.
Rica ederim hocam.
Var mı soru? Bilgi hocam var mı sorunuz?
Hocam yok, teşekkür ederim. Belki burada bir hazır Emine Hanım'ın konusuyla ilgili şimdi son dönemlerde özellikle Amerika'da da bu gıdalarda kullanılan renklendirici katkı maddelerinden bazıları bu kırmızı boya No3, No40 gibi eritrosin gibi bazı renklendiriciler yasaklandı. Bunlar çünkü sentetik oldukları için olumsuz etkileri kanıtlandığı için Amerika yasasına göre yasaklandı. Hatta bu Türkiye'ye de geldi, Avrupa yasası dahilinde. Şimdi o yüzden bu tür çalışmalar doğal kaynaklardan karotenoidlerin renk verici özelliği de olduğu için Emine Hanım'ın bahsettiği gibi doğal kaynaklardan elde edilerek korunması, renk verici materyallerin de bu şekilde korunması aslında daha bir önem kazanmış oluyor. Bu yöntemlerin kullanılarak enkapsüle edilmesiyle. Benim burada naçizane son dönemde yaşanan bilgilendirmeyi sizinle de paylaşmak istedim. Tam da yeri gelmiş diye
Teşekkür
düşündüm.
ederiz.
Teşekkürler Emine Hanım'a.
Sağ
Rica
olun.
ederim hocam.
Emine Hanım teşekkür ederiz.
Ben teşekkür ederim hocam.
If there are no other questions, we will continue with Abdul Bari Hejran. Abdul Bari.
Hello, hello everyone in host. We are going to present our presentation in
Yes.
Mr. Saken Khaidarov taking the lead to deliver the presentation. We sincerely appreciate your time and attentive consideration.
Yes, you may share your presentation.
Okay. So do you hear me?
Yes, we can hear you.
Sure.
Okay, thank you. So thank you very much that you invited me in such wonderful conference. My topic today was going to be bacteriophage therapy for Carbapenem-Resistant Acinetobacter baumannii, a novel approach for drug treatment and drug-resistant infections. I'm doing it on behalf of Abdul Bari Hejran and Parwiz Niazi. So let's have a look what's actually the bacteriophage is. Bacteriophage is highly specific viruses that attack bacteria only. In our case, if we will talk about our strain, we will take it as crab. So it's quite a widespread health threat to the people who get infected because antibiotics are helpless during this infection period. If we talk about the phage therapy, we always have to take into account that we must be specified and can synergize these approaches together with other traditional ways. What's actually the main problem of this treatment is that bacteria, when they develop resistance, they form biofilms, and these biofilms are not permeable to the chemicals like antibiotic that can seriously disrupt their translation mechanisms. Clinical success stories, it's actually a wide-known fact that bacteriophages are going to be effective both on animal models and case studies in clinics. I also would like to light out the challenges in phage therapy because we still have to do a lot of work, especially in terms of resistant development, because not all drugs react equally. Also, immune response, especially in terms of primary immune response or innate immune response with hormones inside them. And of course, regulatory hurdles that any country or any kind of lab can have during these experiments. What could be done with innovations in this direction? So we can do cocktails too and tailor phages, which we're going to discuss further. Nanotechnology and phage delivery is also one of the greatest directions that can make our medical treatments of drug-resistant pathogens, not only bacteria, it could be also some other forms like protistas, one-cell organisms. Advanced delivery systems like liposomes improve phage stability and precision treatment. CRISPR-Cas systems is also quite interesting stuff because it can destroy the gene that responsible for drug resistance, which also quite promising direction. Global collaboration here is very important because the more data we get, the less mistakes we can do in the future. So there is an overview. So let's go to the abstract. So as I said before, bacteriophages is actually the second chance if we don't have effective antibiotics, especially if we talk about the first gen antibiotics. So what is CRAP actually? CRAP is listed by World Health Organization as a critical priority pathogen due to escalating, sorry, I can't hear, escalating antibiotic resistance. It causes severe non-somatical and community-acquired infections, often in ICUs because we get these strains, especially in the hospitalized environments. That's actually the great strategy of this. Then we have biofilm formation and resistance gene acquisition make infections difficult to treat in terms of adaptation period. We know that bacterial infections could be quite fast and they require immediate response because within several days or even hours can be decisive. Phage display, phage therapy, I would like also to add one additional information that it was mostly developed in Soviet countries, and now it was actually initially developed in Soviet Union and then widely spread in post-Soviet countries because maybe you know that Soviet Union had serious problems with antibiotics and modern medicine at that time had to find a way to find alternatives against effective antibiotics. Then we have to demonstrate the synergic effects and the potential of phage therapy that can lead to many options in treatment and can see in other directions. So for example, it's a very interesting table. Why? Because we can see that there is a huge span of concentration of well-known antibiotics that can be used against these various strains of CRAP, as you can see. So the most effective is 400 microgram per milliliter and 256. It's quite huge. So the toxicity effect is extremely high and selectivity index is very low. So this is that it could be actually ignored as therapeutics. But here you see if we combine with phages, we can achieve maximum therapeutic effect if we reduce actually the initial concentration of the drug in terms of antibiotics. Effectiveness of phage therapy in clinical or preclinical studies is very important. Why? Because preclinical studies actually is a pre-phase for everything. Mostly it is made on mice, wild-type mice, humanized mice, transgenic mice, and other type of, I wouldn't say primitive mammals, but mammals that can have any kind of relations to us in terms of effectiveness, of course. Then critically ill patient also can be a target of this preclinical test because they have nothing to lose because extremely resistant strain of this bacteria can kill any person, I mean, severely infected person within hours, not even days. Similarly, a study involving pan-drug-resistant crab strain showed that the phage, this is the name, effectively impeded biofilm formation. Why it's very important not to form the biofilm? Biofilm it's a sign of effective colonization. So bacteria form colonies in order to be resistant not only against antibiotics, but also to be effective against ever-changing environment. I also would like to mention that phage antibiotic synergy improves antimicrobial activity and reduces resistance. It's like a two-sided sword. So on the one hand, you are seriously fighting with bacterial replication. On the other hand, you are making resistant capacities or potential of the bacteria quite weaker because there is nothing to be adapted to. The antibiotics actually is a chemical that do not change, whereas phage displaces changes, and they do have this potential, and bacteria cannot fight two threats simultaneously. If you see that if we add cryptofloxacin or meropenem, improved survival in infection models, then the phage YC06 plus antibiotics suppress the biofilms and reduce required antibiotics doses. PAC therapy offers a promising solution of MDR. Why it's very interesting? Because MDR is also a great tool for those who have multiple infections, not only the crab itself. And you can see in this diagram how many options we do have here. The next table also shows us the so-called molecular properties. You see this GC content is very important. It's a purine content, and it tells us that phages are unfortunately quite unstable. So phages are not long-lasting therapy. They must be renewed at least 3 times during the prescribed therapy. We also can see that genome is quite small. I would say that in some extent it's, I mean, in comparison, of course, to bacteria, it's small, but in comparison to, for example, classical viruses like SARS-CoV-2, it's big. Then major protein sizes were also very important because you can see there is not so big, I would say, differences, but in some extent it could be crucial. Taxonomy and characteristics. What actually our guest is? He is Entombacter baumannii, is a gram-negative, obligatory aerobic, non-fermentative, non-motile, catalysis positive, and oxidase negative bacterium. It belongs to the Acinetobacter Colocacetius baumannii complex, which also encompasses A. colocactus and Acinetobacter genomic species 13TU. This bacteria is predominantly found in soil and aquatic environments. But I also would say that major driver for indiscriminate antibiotic use is rise of MDR and XDR strains. Resistance through B lactamase efflux pumps membrane permanently changes. The changes that we always see in strain development, I mean, in the strains that develop drug resistance, are showing high throughout, not only in this efflux of pumps, but also you can see entire metabolism get acquired. About 90% of isolate resistance to multiple antibiotic classes. Colistin is often the last effective option. but I would say it shouldn't be the last call because colistin resistance can be developed very fast. Horizontal gene transfer aids resistance spread. For example, resistance variants exhibited in marked increase in the frequency of only 8 base pairs, so you can imagine how small is this. And about 70% of lemonade resistant and 62% would be enough to develop this kind of resistant strains, which is actually very troubling for us. Here we can also see where we can find it and what kind of animals could be affected by this disease, and then of course transferred to the humans. So it's actually, there is no need to read out. What's also very interesting is that in table 4 we see that where it can be found mostly. Yes, so mostly interesting that urinary tract infections also can be accompanied with this nasty bacteria. Of course, for me, as a specialist in virology, pneumonia is a main enemy, because pneumonia is only then get activated when co-infection appears. So in this case, leading isolated pneumonia till 96%, which is very alarming, and septicemia is also 90%, quite high. If we see the process of so-called pathogenesis, here we can see that a critical mass, so-called antigen critical mass, must be gained in order primary immune system or innate immune system can be activated. So when the primary immune system starts a sepsis syndrome, they activate all primary immune dependent, I would say also, how to say, these immune hormones that get activated in the first line. Two key replication pathways phages, lytic cycle. What is it? Phage attaches to bacteria and injects DNA. Interestingly, that these phages, what we are talking about, they are DNA attached and mostly double-stranded, whereas most phages prefer, if we can say that word, prefer mRNA.
Professor, you have 5 last minutes, please.
Yes, okay, sorry. Then we see
5
these
last
mechanisms.
minutes.
Yes. Then clinical studies, 88 years old patient with CRAP-induced pneumonia had a marked recovery by inhaling this lytic phage therapy. And actually, in general, if you talk about that, mortality is about 30%, but with this treatment, it can be reduced till 17% subsequently. Then you can see here the graph. We can see that control has this odd values 1. Phage only, it's almost double effect. Antibiotic only has no effect. And phage plus antibiotic has also, in form of biofilm density, is quite ineffective in terms of treatment. So it must be a combination of the treatment. Here we can also say we have 4 different phages. Directed evolution has yielded CRAP-specific phages. Yes, so it's actually limits the stability of the genome. Yes, you see that the yellow one is most variant, and the gray one is most stable. Standardization in phage formulations is also quite a great purpose, because develop unified protocols for purification, storage, and delivery. It's very important, and adheres to GMP standards for safety and reproductivity. It's also very important to understand that phage libraries must be done and must be updated all the time, because phages are keen to, how to say, to change their genome all the time. What we also have to take into account that advance in therapy strategies is actually the main purpose of this direction, because engineered phages and phage cocktails improve treatment efficacy and reduce resistance development. Here's used materials, and thank you for your time and attention. The questions could be asked also to my colleagues which are attending with me. Thank you very much.
Thank you for the presentation.
You're welcome.
Are there any questions with the presentation? May I ask a question, please?
Yes, you can ask Hejran. Maybe he knows better.
Yes, maybe Hejran or you, I don't know.
Mhm.
Anyone may answer. I'm not in this area. I'm not working in this area, but I wonder, are there any risks related with using this phage method? Yeah, I think you use viruses against bacteria. Am I right?
Yes, I think so. I will try to answer me first, and then Hejran will take ahead. Okay. So I would say you shouldn't be alarmed because they're highly specific, and they attack bacteria only. The only thing that could be under the risk that they can attack only positive bacteria that in your stomach or in your intestine. Yes, that's only the risk that you can have. So they are not specialized in human tissue and the human cells. They're not interested.
Okay.
And also they have DNA in their main genome, so it's quite ineffective.
Okay, okay, thank you.
You're welcome.
Any other questions? So let's continue with Züleyha.
Thank you
Are
very
you
much.
ready? The next presenter is Züleyha Muhtar.
Assalamu alaikum. Good evening, everyone.
Walaikum salam. Yes, and
I'm Zuleyha Girat Mukhtar. On behalf of my co-authors, I would like to appreciate and thank you for the opportunity to share our study on this platform. So our presentation is titled Hibiscus subdrafifa Extracts as Natural Alternatives for Food Preservation. So this is a preliminary study on antioxidant and antimicrobial activities of hibiscus flower. So the next slide contains the outline that I'm going to be guided for, starting from introduction, results, materials and methods, discussion, conclusion, and then the recommendations. So this actually is a preliminary study we started with a colleague and some of my students, and it stemmed from our curiosity regarding the hibiscus flower. So it's a common plant for here, us in Kano, Nigeria, northern part of Nigeria in Africa. And what we use it to make drinks and tea. And we observe that it is one of the few commodities that if you keep for a long time, despite the weather here where it's quite hot and humid, you will notice that it's quite resistant to mold. So based on that, we got curious and said, what if we can use the extract as a natural preservative? Being people are more health conscious and want clean label products, and the consistent concerns over synthetic preservatives such as benzoates and sulfites, it seems an interesting use for this plant that we have, especially because it will be cheaper and it is available even if it is in small quantities. So it is also one plant that from the literature review we observed is very, very rich in phytochemicals such as anthocyanins, flavonoids, and organic acids. And we all know flavonoids are very, very potent antioxidant phytochemicals. So as I said, it is well established. It's well established uses as a drink and tea. And then we are having some health benefits because some researchers have shown that it lowers blood pressure and it is also good for diabetics. But this application as a natural preservative is really generally underexplored. So this research aims to develop and evaluate its natural preservative potential. So next slide. So we sampled the hibiscus flowers from different areas in the farms here in Kano. So they were authenticated and prepared it by, we prepared the sample by washing it, drying it, and extracting it using two methods. So next slide, please. So for the extraction process, we use two extraction methods. Next slide. We use ethanol to extract it, and then we used we also got the aqueous extract so that we can compare which is better. So next slide. Next, please. So we also the next slide. So we also conducted phytochemical screening, where we did qualitative and quantitative analysis of flavonoids, anthocyanins, tannins, phenols, and saponins according to standard methods, so that we can have an idea of the quantity and of these potent antioxidant compounds. And we also conducted an antioxidant activity assay using DPPH free radical scavenging assay. Next slide, please. We also assessed the extract's antimicrobial activity against some commonly known food spoilage microorganisms such as E. coli, fungi, the fungi Aspergillus niger, Salmonella, and Staphylococcus aureus. The next slide. So we also tested the extract on some common food products. So we tested on juice, tomato sauce, bread, and fresh milk. So what we did is we divided them into three groups. One group was treated with the ethanolic extract. The second group was treated with the aqueous extract, sorry. And then we left one group as the untreated control. Next group, please. Next slide, sorry. So we also conducted statistical analysis on the results we generated. Next slide. So we have the result of the phytochemical analysis, and it shows that the ethanolic extract showed higher concentration of the phytochemicals tested for. The aqueous extract, even though it contained all the phytochemicals, showed a lower concentration in relation to the ethanolic extract, meaning that the ethanol served as a better, the ethanolic method served as a better extraction method for the extract. Next slide. So for the antioxidant activity, the ethanolic extract exhibited a higher free radical scavenging activity compared to the aqueous extract. So this shows that it is also going to be a better preservative in the extract we got using the aqueous extraction method. So for the this table shows the results for the antimicrobial activity. Next slide. So this is the table for the food preservation trials. As you can see from the table, the first column is for the control, the second one for the ethanolic extract, and the third for the aqueous extract. So the number of days is correlated to the number of the time frame after which it started to spoil. So based on this, you can see that it was quite effective and definitely better than the control. For the control, for example, with the pineapple juice, 7 days at ambient temperature, 10 days in the refrigerator. But with the ethanolic extract, it was 18 days and 21 days. And then with the aqueous extract, it was 15 days and 18 days. Same with the bread, same with the tomato sauce, and same with the fresh milk. So next slide, please. So this data strongly supports the hypothesis that hibiscus flower extract particularly the ethanolic extract, possesses considerable antioxidant and antimicrobial properties. Based on the results we got, that is the quantitative phytochemical screening, it is definitely rich, notably in flavonoids, which are very, very good and strong, potent antioxidant compounds, which are critical in delaying oxidative degradation and microbial spoilage in diverse food systems. And it also showed notable effectiveness against both gram-positive and gram-negative bacteria, fungi, and yeast. So this broad spectrum action is also a very positive thing, as it means it can be used on a wide variety of microorganisms. So, and the food application trials with the 4 diverse food matrices demonstrated significant extension of the shelf life. So this is very important, especially for us here in Africa, where there's a lot of wastage from the farm and either in our houses because of instability of light and all these factors. So it serves as a very cheap alternative way to preserve our foods. In conclusion, this study successfully showed that hibiscus flower can serve as a natural preservative. It demonstrated strong antioxidant and antimicrobial properties and effectively prolonged the shelf life of the different food products tested in this trial. So this holds promise for reducing food spoilage and meeting consumer and definitely local demand for natural preservatives. So the next slide, please. So we recommend conducting trials on a larger scale and maybe nano-encapsulation toxicology studies to assess the acute and chronic toxicity levels for it to meet standards for food additives, and then maybe developing commercial food products fortified with this extract to promote its use. So thank you very, very much for listening, and thank you for giving us this the opportunity to present our findings. Thank you.
Thank you for the presentation, Dr. Züleyha Cidado. Does anyone have any questions?
Can I? I will have only few questions about the study, if it is possible.
Yes, sure.
In the study, you mentioned about after you get the extracts, you applied those extracts to the beverages like milk, fresh milk, bakery, and some kind of other
bread,
juices. Yeah. So what was the form of the extract? Was it in a powder form, or did you apply it as a liquid? And after the application, did you see any effects on the color or any other sensorial qualities of the final product? I mean, did you
with so this hibiscus flower has a very, very strong, deep red
that's
color.
the reason I asked that question.
Yes. So like I said, this is a preliminary study to test whether it works or not. So definitely there were physical changes to the samples. So like we used the extract, and for like the bread we used the dried powder. But at this level, that is why we recommended maybe we can remove the color, or you can use it we can maybe test in future if we can just get the extract, remove the color, remove the flavor, remove and get it in a form that you can use it without affecting what you have. Like for the fresh milk, it changed it to pink color. That is not desirable, but then the effect was there. It actually preserved it.
But if you present it into markets as a colored milk or milk with high antioxidants, maybe it may find a good place for it, huh? For the
okay, we'll try that.
Okay,
Because
thank you.
at the moment, it's one of the things we're looking at. The color and the flavor is there and it is very, very strong. So how do you convert it in a form that you can use it as a preservative without affecting your own product?
Okay. Thank you. Thank you for your
Thank you very much.
nice work study. Thank you.
Any other questions? Then Maman Çapıyor. Maman Çapıyor.
Yes, sir.
You may
Yes, sir.
start.
Yeah.
Halil Ünver burada mı? Burada galiba.
Sir, I'm waiting for this.
Yes, Maman, we are waiting for you.
I can share my
Yes,
PowerPoint.
yes, you can start, Maman.
Well, thank you very much.
We also thank you, Züleyha.
Okay, sir. I didn't see how can. Yes, yes, yes. I saw it. So I will start. Okay. Let's go. Sure. Sorry. Hi everyone. Hi everyone. Sir, may I start?
Yes, please.
Okay. Hi everyone. My name is Muhammad. As a professor said, Muhammad Chapi Sulegarba. I'm from Niger. So thank you for the invitation. So it's great to present our work. So the work is entitled Revalorization Iron Copper or Mining Tailing by Flotation. So
Can you make it
as sir,
full screen, please? Full screen, tam ekran.
it's okay. Okay.
Yes, it's okay now.
Okay. So as I said, this the title is Revalorization Iron Copper or Mining Tailing by Flotation. So as any works, they have those presentation outlets. So we will start by the introduction. So giving an overview of tailings, study of areas, of course, maybe some of the participants, they don't know what's the tailings. Then study area, objectives, matrix and methods, result and discussion, and the end we conclude by presenting the results and some recommendation. So as I said, so from the introduction, so you know, mining and mineral processing, so they are producing a large quantities of tailings. So from mineral processing, as you see the image here. So from here you have the inputs. We call it the feed. So this is the method flotation process. There are many separation methods, but flotation is the most effective techniques which use the physico-chemical properties of the minerals. So after the separation, you are getting two products, concentrate and tailings. So the concentrate is the final product or the products which contain valuable minerals. And the tailings are the waste and non-valuable minerals. But of course, it's not 100% waste because even in the tailings, you will find valuable minerals, because in the processing you can't process 100%. Always you lost some valuable minerals. So the flotation, as I said, is the most effective techniques which is used in mineral processing. So which area is used? This is used for base materials, precious metals, gold, silvers, and industrial minerals like phosphates, feldsphates, and so on. So as I said, the mining and mineral processing, they are producing millions of tailings. So from not only he said 10 millions of tailings were produced, whereas 230 millions were produced from gold. Why from gold? Because when you take a minerals, a gold ore, from 1 ton, you are getting only 5 grams. So that's why you are getting many waste. So many quantities of waste. For the other metals, you are getting kilogram instead of gram. So this is the graph that show the tailings which produce in some countries. You see the difference in Africa is very low, below 50 million. So West, you have difference. It can come from gravity processes, flotation process, or leaching process. You see from the image, you see the storage as mining waste storage. So this is the picture of underground mining and open pit mining. You see, so from how can you get a tailings? You see from the figures, you have the ore. After the ore, the extraction, you get mining waste. And then from the separation, you are getting tailings. And for metallurgical process, using high temperatures, so you are calling slags. So at the end, you get the metals as valuable minerals. See here, there are different type of tailings. It can be liquid, gas, and solid. So how the ore and other minerals appear in the process? This is an example. As I told you, you can't get 100%. You see, from here, the concentrate here, the concentrate here, you have first the rafa, you have the mix of the gang of tailings, as we call it. At the end, you are getting the final product, we call it concentrate. You see, it's liberated. You see, so now money, they are facing problem because you can't find high grade of ore. So as so many plants, many mining companies, they are expecting low grade of mining. So how can we process this type of ores or tailings? Is by characterization. If you do a characterization, so you get the information. Why the information is about the mineralogical properties, physico-chemical properties of the minerals, which allows us to choose the methods. So that's why this is our study area in Bayakı, where we took the sample the samples to do is the project we made in 2021. You see here. So this is the flotation units in mining, iron mining, iron copper flotation plants. You see. So from why sometimes the mining company are giving projects is to find a solution. So we, as I said before, we are producing millions of tons of tailings. So these tailings we can't left it. We can use it for another things. It can be used as a marketable product. So from the characterization, we can find a solution about that. So the objectives of our study is to perform the mechanical process, performing mineralogical analysis. Why? Because to
Dr.
determine,
Muhammad,
of course, sir.
Dr. Muhammad, you have 5 minutes.
5 minutes, okay.
Yes.
Conducting the laboratory flotation, carry out the economical analysis. So here is the sample. In mineral processing, the very important thing is how to take the samples. So it's very important because if you can't get a wrong samples, you can't get a good result. You see here as we prepared, because there is some humidity during moisture contents, so we have to dry it, grind it, and use to get a representative samples. You see here. So this is represented. So we take 1 of 4 to make our team our analysis. So here is size distribution analysis we made. So the moisture contents. You see here is the chemical analysis we do. Before we have to prepare the sample and to digest the samples to get a solution. Then we bring it to our ICP to determine the minerals contents. Then and also we use for XRD to determine mineralogical composition. Then the chemical composition we use also the X-ray fluorescence spectrometer. So here this is our flotation test we made to recover our valuable minerals which we call molybdenum. So this is the different procedures we made. We took at start is basic 7.5. We add NaOH. So for the solid ratio is about 20%. So for the condition time, it's very important because to mix the volume minerals and others and the solution water. So then flotation time is about 3 minutes. So we made around three tests, and then we get result. So here after we get the concentrate, so we bring it to vacuum to dry it to get our concentrate, to which we will do another chemical analysis to determine the metals content in our concentrates. So here is the flow sheet we made, because in our study we made laboratory tests and also economical analysis to estimate the real cost of operating costs, because the money storage the tailings. So the solution we are bringing is about to bring adding another unit of flotation to recover the molybdenum. this is our flow sheet. So for the economical analysis, we start for developing the flow sheet, list any size of corresponding equipment, determine the capital cost, and at the end is the operating cost. You see here is our flotation, or the flow sheet of our flotation. So for the result, coming for the result, you see here for microscope, we get the chalcopyrites, magnetics, and pyrites. And from the numerical analysis, we get around the same minerals, chalcopyrites, dianites, and magnetites. So for the chemical analysis we made, because we made a size distribution analysis for each size, we have take the weight and also made the chemical analysis to determine the metal content. We see from here, because the ore is, our tailings is iron copper. We see iron has more percentage comparing to other metals. So here is our chemical analysis, our explorations analysis to determine the chemical analysis, the chemical composition of our samples. So the result also we had is corresponding with the other works like Autoimmune, Quran, and Abdul. So here the chemical analysis. So here for our the flotation result, we see here we are over 78% of our molybdenum with grade 11.78, which is 60. So the important thing is what? When we are using more concentration or more quantities of an NISH, we are getting more recovery. So when we add like 1,000 of NISH, we are getting like 81.60% of our molybdenum. So from the economic analysis, this is what we started. So we see here we have the feed and also the equipment we are estimating. Here you see here for the price, because they are going to establish and process a flotation unit in their company. So this is the complete feasibility. In the complete feasibility, we are taking for like for the years 15 years. So how we can estimate the net present value, maximum capital risk, benefit cost, internal rate of return. So all the tests we made, so we are seeing they can process this type of tailings with profit, because in mining you should process an ore with profit. So we are coming to the conclusion here. The mineral composition, we see the iron is abundant according to the particle size. We have more concentration. You see 13.58% is around more than 135 kilogram by ton. You see, so the flotation results showed that successful molecular recovery with 98%, of course, and the economical provide an estimation cost of the equipment and operating cost for molecular at industrial scale. So what I'm going to say, so the economic analysis is very important to the industrials to establish and flotation or also to establish a plant. So that's why it's very important to make the economic analysis to our project to estimate the cost doing the operators. So coming to this is my references. Our references we used. We see. So if there is any question, I'm glad to hear you.
Thank you,
Thank you.
Doctor,
Yes.
for
Yes.
the
Sir.
presentation.
Yes. Sir.
So we may take the questions.
Yes.
Soru
Yes. Sir.
soran var mı arkadaşlar? I think since this is the last session, really last session because the others finished, I think
Sir,
nobody questions. So thank
sorry,
you.
I have another question. I have another presentation.
So, but we will continue with Halil Ünver, and after him, you will make your second presentation, okay?
Thank you, sir. Thank you, sir.
Okay. Can you stop sharing? Muhammad, can you stop sharing presentation?
Tamam.
Yes. Halil, you may share your presentation.
Yeah, hey everyone. Can you hear me?
Yes, we can hear you,
Okay.
and we can see presentation.
Firstly, thank you for the opportunity. I am Halil Ünver, a second year student in mathematics at Ege University. And my study is a mathematical approach to a green university for a more sustainable future. Universities have direct and indirect effects on the environment due to the large population and various activities they host, and for the reason they are called small cities in the lecturer. Based on this definition, we thought of faculties and neighborhoods of these cities and departments as a street of these neighborhoods. Why universities? Because in sustainability movements, the transformative power of universities is felt not only through education, but also through being role models. With this project, we investigated how this chain transformation at the university level could be possible, starting from small units. And as you can see, our hypothesis in this study, in fact, I already said it, demonstrating the possibility of a green future at the department level within universities that could lead to the feasibility of a green faculty. The feasibility of a green faculty could contribute to the creation of a greener university, and a greener university could give rise to a greener society. Our aim in the project is to seek answer to these two main questions. First, what are the tendencies of the participants regarding the reduction of the ecological footprints in terms of food, transportation and shelter, energy, waste, and water consumption? And second one is, does the ecological footprints awarenesses of the participants, along with the tendency in the sub-dimensions of this scale, namely food, transportation and shelter, energy, waste, and water consumption, differ significantly according to gender, level of education, department, monthly personal expenses, whether or not they have received environmental training before. We collected data from a total of 277 participants. Our participants were students, academic staff, and employees of the Faculty of Science at Ege University. The survey we used was a 40 question under the 5 sub-dimension: food, transportation and shelter, energy, waste, and water consumption. And we used a 5-point Likert scale. Like absolutely agree, I agree, or I absolutely disagree, to measure attitudes and behaviors. After completing our survey, we first categorized and organized our data. Then we used combust alpha test to test the reliability of survey before starting our statistical analysis. As a result of the test, we achieved a very high level of reliability, more than 90% reliable, and thus we start to conduct our tests. For two group dimensions, I mean two groups, for example, gender, female or male, or environmental education, yes or no. We used independent t-test and determined whether there was a significant differences between the groups according to the result of the applied t-test. In groups of 3 or more, we applied the ANOVA test for the average scores of the participants on the tendency scale. In order for ANOVA to be valid, the variance of the groups must be close to each other. Therefore, we performed the Levene test for ANOVA to test whether the variances between the groups were homogeneous. If the p-value obtained as a result of the Levene test bigger than 0.05, this shows that the variances are homogeneous and the ANOVA results are reliable. If we obtain these results, we produce to the ANOVA test, and if a significant differences was deducted with the ANOVA, we performed a post hoc analysis by applying the Chef test to determine which groups the difference was between. Later, we wanted to support this project with machine learning. For this, we used support vector machine, artificial neural network, random forest, XGBoost, and logistic regression as classification algorithms. And we used Smooth to balance the data and normalize with the standard scalar before modeling. And we used accuracy, sensitivity, specificity, precision, and F1 score to evaluate the predictive performance of the machine learning results. And as a result, we basically solved this. In support of the lecturer, females showed higher environmental sensitivity than males. And we also saw that having received environmental education increased sensitivity. But contrary to the lecturer, we found that people with high monthly expenses in the West subdimension showed lower environmental awarenesses than others. This is a rare situation in the lecturer. Paul Stern, who emphasized this situation in this study published in 2000, said that financial means are not always directly proportional to environmental responsibility awarenesses, and that other factors can also be effective in environmentally friendly behavior. When we conduct an analysis according to subdimension, we observed in food subdimension variables such as gender, education level, department, and environmental education create significant differences between with environmental awarenesses, while monthly expenses does not create significant differences in this dimension. In transportation and shelter, the subdimension, significant differences was observed only in education level. In energy, gender, education level, and environmental education factors created a difference, while education level and monthly expenses do not create a difference. In the West subdimension, education level and monthly expenses factors create a difference. The others do not create a difference. In the water consumption, gender, education level, department, and environmental education create a difference, while monthly expenses does not create a difference. Then in machine learning results, in the accuracy metric, random forest and XGBoost gave the highest accuracy. In the sensitivity metric, most models gave low results. This showed that the class imbalance could not be completely eliminated despite smooth.
Last 5 minutes.
Yes, okay.
Thanks.
In the specificity and precision metrics, random forest and XGBoost gave the highest specificity. The best performance in F1 score was in the logical regression model. Our findings confirmed some trends previously emphasized in the lecture. However, contrary to the lecture, we found that waste management tendency were higher in low and middle income group participants in the waste subdimension. In higher income groups, waste management tendency were relatively lower. The 50% to 65% accuracy rates obtained in the machine learning section relieved that demography variables alone could not adequately predict environmental tendency of participants. And of course, there are some limitation in this study. Some of this limitation, as you can see, our study contains response bias. The sample consists only of members from a university faculty of science, limiting the generalizability of the findings. Only demographic variables were used in machine learning models. Psychological, social, and cultural factors were not included. The machine learning analysis includes only a subset of algorithms: support vector machine, artificial neural network, random forest, XGBoost, and logistic regression. Other modeling approaches were not explored. This method was applied to address class imbalance, but synthetic data may not fully represent the true data distribution, potentially affecting model validity. And this project is supported by the TikTok, so I sincerely thank TikTok for its support. And thank you for attention and for listening.
Thank you for the nice presentation, Halil. It's a really nice study.
Thank you.
You are in computer engineering department? Mathematics.
I couldn't hear my.
Are you in mathematics department
my
or
network,
computer engineering department? Are you in mathematics or computer?
I'm in mathematics.
Okay, that's much better. In your department, you started studying with AI?
Yes.
Are there any questions to Halil Ünver about his presentation? Yes, I think everybody is tired at this hour. And before the last presentation by Mahaman Chapiu, can everybody turn on their cameras and let us take a photo and then continue with the last presentation of Mahaman Chapiu? Yes, Salisu. Salisu is working. Evet, Halil. Dr. Züleyha, can you turn on your camera? Abdul Bari, Hejran, can you turn on your camera, please? Yes, thank you. I'm taking, if you are ready. Salisu, just stop 1 minute. Just stop. Huh? That's okay. That's okay. Just a minute, please. Thank you, everybody. Now we will continue with Mahaman Chapuy, the final presentation of the Congress. Mehmet, it's your turn. You have
Yes, sir.
10 minutes this time.
Thank you, sir. Start. Sorry, sir. Okay. Hi everyone again. So as I was time, my topic is investigating the environmental impact of small-scale mining activities and propose a suitable restoration techniques. So as you said, you have minings, a large-scale mining and small-scale mining. So we give details about small-scale mining. So from the presentation outlet, we start with introduction, start the area objectives and methodology, explain and result and discussion, then conclusion. We end with some basic recommendations. So we see from the artisanal extension, artisanal mining is or small scale mining. It's like small activities which can be done by hand or by mechanical equipment. You see here, this is a picture from one of artisanal mining. So this type of the activities, of course, it's generating an employment, but these activities has many problems because they are producing many of heavy metals or also some pollution to our environment. The committee for today's objectives, we conduct a survey with people who are working for this type of activities, then given the environmental impacts of various mining measure, we determine the most effective and suitable methods for the application, the socio-economic factors, presenting the policy and regulation. Because of this type of activities, there is no regulation. They are doing these activities inappropriately. So from here is our study area is in northern Niger, Abid, which contain large resources. Of course, it's desert, but in this city you have like three mining company, uranium mining. You know, when we are talking about Niger, we are talking about uranium. So that's why. So the potential is like how the resources are in another sources, another country, in another cities. We see here from Damagam, we have lead, antimony, zinc, and gold resource, gold ore. We didn't see, we are not just start yet, but maybe in the future we are going to mine those minerals. So for materials and metals, this is the activities they are doing. When you see here, as I said before, so it's by hand or semi-mechanized. So you see here they are using their hands to crush the rock they are extracted. You see here, and also there's me, but there's no rules. There's no law. Just they are doing it. So get at the end the ore. So here you see Another is for cognition. Cognition, when we are taking neural processing, is mechanical preparation. Mechanical preparation. Why mechanical preparation? Because we have to reduce the size and integrate the valuable minerals. So that's why we are doing cognition. So from here, you have is about the separation, which we are calling is incineration. Incineration is adding the shell, but you see here is like traditional they are using who dissolve the gold metals. You see here is acidity and also they are doing is a traditional, but they are doing in order to get money and do something. So you have here the next step is about the washing, the cleaning. They are cleaning in order to get the air. Then they are using nitric acid also to dissolve. Then they are washing. For example, in mineral processing, we are not doing that. There are equipment who are specialized for this type of work. You will see here, these people are exposed. So, but in order to earn money, they have to do that. So, from the results we made, the sample we made, we took 20% and make an survey. So when you see here, around the age for 36 to 16 to 35, there are more people who are doing this type of work, small scale minings. You see here. Then and also we ask question about the education level. So this type of people you see here around is just 20% who did primary school, secondary school, but when you come into the university, it's around 0%. You see, so what's impacts are those type of activities is environmental impact because they are destroying the environment. You see here to get the gold ore, they are doing some excavation, which there's no norms. They are doing that and they are destroying the source. Another impact is about also the storage. You see here, they are storing the waste and they are living there, which they are exposed to themselves. And they will get sick. They will get sick. Unfortunately, they have to do that to end their lives. So here also the vegetation. So you see the trees are very destroyed, and also here. And coming to the resources, which water is very important.
Doctor,
You see, yes.
you have
Yes, sir.
left 3 minutes.
Okay, I come to the end. So we see here from the resources, this waters, the area surrounding, they are using this type of waters. So it's contaminated, of course, because we get the samples and made some analysis. We see here which we got from different samples. We have the concentration about heavy metals. When we are talking about heavy metals, we are talking the metals which has the density greater than 5 grams per centimeter cube. You see, which is very dangerous for humans, but we took from the samples. And also when it comes from the airs, they are contamination our atmospheric. So you see, so this impact is very, very negative for the environment, but those people, they have to do that because in order to get money or employment, so they have to work that. I remember in 2023, there have dramatic things which happens, which cause death of many people in gold artisanal gold mining. Unfortunately, sharing this type of information, we see here from the atmospheric when we come to preparing from grinding, we see this type of this mud, and also from the mechanic. So they are putting a lot of dust. So coming for the restoration methods, we suggest, because in my thesis we did we use phosphate-based materials, and also we made some analysis after the precipitation. We made again an analysis to see if the metals can dissolve after decreasing the pH. So this method it's very innovative, and also it can be important for the government. It's cheap. Why it's cheap? Phosphate-based material fertilizer, we are using that, and also we can get it everywhere. So that's why we, for the use of phosphate-based materials, we said it's very important to use that in order to restore or rehabilitate this type of mining site. And also the another method is phytoremediation, which is innovative, of course, but it takes time. Why it takes time? Because you are using trees which will take the metals. It takes many years or years, but the use of phosphate-based materials reagents, it precipitates the heavy metals, which even in acid medium, they can't dissolve. This is an example of one project about the remediation methods. Then, to the end, so the calls was today has significant orange contents that affects has become one of the mining sector where environmental powers are most worried. So, in order that we recommend the government can more involved in this sector by regulating operating techniques, minimizing negative social, economic, and environmental impact as much as possible. So, this is our references. Thank you for your listening. If there is any question, I'm glad to hear that. Thank you.
Thank you, Doctor.
Yes, Dr. Muhammad Chapiv.
Yes, sir.
It was a very pleasant presentation. And with this presentation, we conclude our Congress.
Yes, thank you, sir. Thank you, sir.
There is only one person that might ask you a question. Alif Diambu. Alif, do you have a question?
No, I don't have. It was clear, and we are already tired now.
Yes, sir. Even me and
Chapiv.
yes, sir. Yes, sir.
We are pleased to see you.
Thank you, dear.
Inshallah.
Inshallah, we'll come next year.
Again.